we go. So my hope is we can manage this to where that this video will do what's important on the whole part of 10-1. So that's 1, 2, 3, pretty much almost 3.5, kind of almost 4 strips. So that would be wonderful. So we're going to do it a little bit differently. So there's this person on YouTube who teaches by drawing, which she's much better than me. So I'm going to watch it with you in this video. And so I'll pause her, we'll look at what it looks like in our notebook, and we'll see if that works. And if it's messy, then I'll just delete this and you'll never know what happened. Okay, so here we go. Okay, so we're going to watch this video together. Let's see how it goes.
Before we have a look at the process of photosynthesis, we need to know what the chloroplast looks like and the structure of it, same as in mitochondria, where they are both originally their own.
I'm going to draw that in my notebook. So I'm just going to, this is going to be quite the trick to manage all of this. I'm going to just go to, we're going to need several pages. It's not going to look like, where'd it go? This guy, this whopper. We're going to scale it back a little bit. But I just need, I'm guessing I'll need 3 really good pages. And like we did before, when we did do the crazy drawing, let's budget ourselves to where that our drawing is no more than 2/3, so we have room to go down here and to write the important information, because, you know, drawings are great, but you need to know what you drew. So I'm going to 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. I'm going to leave about 11 lines. And I know it's just so loud when I move that. I hate it because the microphone's actually part of this guy. So I'm just going to count up 11 from the bottom. 10, 11. So whatever she's drawing, I'm going to only draw above this line. And then I'll come back and write any notes to myself. So, yet again, let's go back. Maybe I can get good at this. I'm kind of doubtful. So, this is what I'm drawing. I won't enlarge it right now. And she does write all around the edge. So, I don't want to make that chloroplast huge. So, I think what I'll do is I'll pause and I'm going to move that to my drawing. And then when I come back, I'll show you my drawing. Okay? Want to use your time. So you may just want to pause too and draw that in your notebook. So that's what I drew. I drew it from where I paused her video, and I'm going to write just some general pieces of information. And here's what I want to include just because it comes from our notebook and our glue ends. So instead of using this chloroplast, so I'm 10.1 number 1, I'm going to go ahead and put that right there. So I know that this came, whoops, let's get that down so you can see. This came from a glue end. I'll put what this is. So this is a chloroplast. And so we're really just wanting to understand the structure, and we'll let her continue with her discussion, and we'll label. I know that she had already said that this piece right here is DNA. Now, a chloroplast is an organelle in a eukaryotic cell. So let's put that up here. You know, I'm lazy, so I'm going to go there. associated with plants. It will also have a drawing that shows where they're located in like a leaf. So one thing I do want to say about the way she's drawn this is it's hard to tell what its actual structure looks like. So I'm going to just put a little note here. I don't think that she does. But the DNA in a chloroplast is circular, like bacterial DNA, like prokaryotes. And the reason is, we have that whole eukaryotic process of absorbing at some point way a long time ago, prokaryotic cells like this chloroplast and the mitochondria. And in that process called endosymbiotic theory, that's the belief that as they took in these and did not digest these different cells, chloroplasts and mitochondria, then you developed the specialization that eukaryotic cells have. But these were once free living. So chloroplasts and mitochondria were once free living. So we want to come back to that theory once we are through drawing this, and we'll highlight and write a little note down here about what supports that theory. Okay, so let's go let her finish. No. Go away. Okay, so here we go. Everything is super slow on this computer. I hope it's doing okay. Come on, I can't even move my mouse. Keep going.
Individual bacteria which got engulfed by the cell in prehistoric ages. As you can see, there are starch grains as well. Photosynthesis is the making of glucose molecules from light energy, and after glucose has been made, they are combined together to make starch and are temporarily stored here. Also, we have ribosomes here, which are all the individual dots. You can see it all across, and they are there for protein synthesis. Photosynthesis is a very complicated biochemical process which requires lots of enzymes to help mediate the reactions. So ribosomes are really important here to make those enzymes for those processes. And DNA here is coding for these enzymes as well. So now we have a look at the sites of specific stages in photosynthesis, namely the light-dependent stage and the light-independent stage. So as you can see here, there are some structures that are like stacks of discs, and these individual discs are what we call thylakoid. A thylakoid, or specifically the thylakoid membrane, is the site for the light-dependent stage, which is what we call Z-scheme as well. So thylakoid can be stacked up as what we call a grana, or in plural form a grana, and they can be connected by the intergranal lamella. And finally we got stroma, which is a fluid that fills up the space within the chloroplast. It's kind of like cytoplasm in a cell, but it's not. It doesn't do the same thing. And stroma is the site for the light-independent stage, which is the second stage of photosynthesis. I also call this light-independent stage Calvin cycle, but we'll go into more detail in another video. Now, a little bit more about the thylakoid membrane structure. In the process of photosynthesis, we need to know by the cell in.
This is not going to be easy.
They are there for protein synthesis.
Where were we?
Look at this can be connected by that scheme. It is, like we said, the light-dependent stage. So on the thylakoid membrane, there are actually structures called photosystems, and they.
Okay, sorry. Whew, all the buttons are laying on top of each other. So what I'm going to do now is I'm going to just show you what I've done. Let me add. I'll pause and just add all that she has on there. Okay, so here's what I have so far. I have that we have a chloroplast. Chloroplasts are now organelles in eukaryotic cells in plants. One thing she did not label is that you're going to have this ability to have a gradient because you have this membrane, and then you've got membranes that are also part of the thylakoid. And so you can have different compartments. And so this is going to be the outer membrane, And then you've got each one of these that look like a little plate, and is called a thylakoid. And in the membrane of the thylakoid is where that light absorption takes place. And that light is used to excite electrons to create high energy electrons that we needed for glucose, which we've already talked about in that previous video. So we just added here that the thylakoid is where the light dependent reactions take place. She calls them the Z reactions. In the US, you're going to hear they're called light dependent reactions. They're located in the thylakoid membrane because the photosystems have to be localized in a membrane, just like the electron transport chain did in cellular respiration. So they're in the membrane of the thylakoid. When you have several thylakoids stacked together, that's called a granum. And when you have a bunch of stacks, those are called grana. And this little bridge that connects them, that's called intergranal lamella. Also, the designation of this middle stuff, just all of the stuff that fills up the chloroplast, is the stroma. And then inside the stroma, you'll find DNA, and this DNA has specific genes and that's going to be 4 processes. So they're actually going to be genes that code for proteins that are used just in the chloroplast. So specific genes for processes in the chloroplast. So it carries its own genes for its own processes. And then the information from the DNA will be transcribed into messenger RNA. Messenger RNA goes to the ribosomes, goes through translation into a protein, and you end up with those proteins that you need to do the work of a chloroplast. So it's got its own specific. And so that takes care of it. The only thing that we didn't say, even though she started off that way, is the purpose of a chloroplast is not to make sugar for you. It is to store energy for the plant. And so first a simple sugar, actually a sugar precursor molecule, we talked about that, like the little Swiss Army knife. And it can be made into many other different types of molecules, the most common one being glucose. And glucose put together in a long chain is a carbohydrate that would be a polysaccharide, and the example that she's using would be starch. And so you've got these little starch storage spots inside there. So let's see what else she has to say and what we need to add. I don't know how this is going. I hope this is okay for you.
Systems 1 and 2 on membrane. And these photosystems are made up of the photosynthetic pigments, which are these structures within these photosystems that absorb the light. We've got the primary pigment and the accessory pigment. And the primary pigment will be the key one that absorbs the sunlight, so it's the main functioning one. You're very familiar with this, which is chlorophyll A. As for accessory pigments, you can have chlorophyll B, xanthophylls, and carotenoids, which are kind of like supplements. They help with the process of absorbing the light energy when chlorophyll A is not really functioning well, but the key one there is the primary pigment chlorophyll A. So a very quick summary, this is the structure of the chloroplast. We've got the DNA, which codes for the enzymes needed for the photosynthesis reaction. We've got the ribosomes that actually make them, and starch as an energy storage. Then we got the thylakoid membrane, which contains photosystems, which contains the photosynthetic pigments like chlorophyll to do the light dependent stage, which is what we call the Z scheme as well. And they can be combined together to form a structure called granum. And grana are connected by the intergranal lamella. And then after this reaction occurs, the substrates then go into the stroma, where light independent stage occurs. We also call this stage the Kelvin cycle. And in our next video,
Okay, so that's good. So now we're done with this whole drawing. Let me go back to what I added. That's weird. Everything's weird today. It's snowing. It wasn't supposed to. There's all kinds of craziness. Okay, so here's what I ended up with. The only thing I've added since you saw it is I added the information that over here in the thylakoid membrane with these light-dependent reactions, those are composed of two photosystems, photosystem 1 and 2. And then I just wrote what pigments there would be. Chlorophyll B, chlorophyll A, accessory pigments. We'll get into that when we get into the light-dependent reactions. So I think what I'm going to do is I'm going to go here and we're going to save this where can chloroplasts be found. We're skipping 2. I think I showed you this already. So we've labeled a chloroplast, a lot more detail than what this would have had. Skipping 2 about emergent properties. Looking at plants where that chloroplast can be found. We'll add that from another video. And what we'll do is we'll either take this cross section and glue it in, and then we'll be able to show. Then we've already written the equations for photosynthesis. And we did that when we were arguing that it's just the inverse of cellular respiration. This talks about gross, which has water on both sides of the equation. That's a little bit beyond you. They don't really ask about that. So in this particular case, they just want you to know what the equation is. Then we get into the light reactions and how that we split water to get the electrons, and then what do those look like. So I think what I'm going to do is stop this one, and this is going to be then, let's be real clear, my budget's 47 minutes, so I have 30 more minutes. So this is part 1 of probably 3 videos, okay? But I'll load them into Edpuzzle one at a time, so hopefully if it kicks you out, then you won't have got so much to start with. So part 1, we just finished. We're going to start part 2, which is the light reactions.